Thank you very much for the opportunity to present virtually. I'm very sorry I couldn't be there in person. It sounds like a wonderful meeting, and all of the talks so far have been really inspiring. So I'll be presenting a short overview of our activities in the past year. I'm the chair of the Image Informatics. My home is at the University of Minnesota at the Supercomputing Institute, where I direct the Research Informatics group. First of all, I wanted to say that we are happy to welcome a few new members. One of them you've just heard speaking, so we're very happy to have Virginie, John, Eric, and Sarah also joined us this year. We still have our primary focus around lowering barriers to learning and communicating image informatics or bioimage informatics. And we meet monthly to connect, exchange news, and also make progress on the various groups' initiatives. So here are a few highlights. First of all, after the last community congress, Beth organized a mini symposium in deep learning for bioimage analysis. That was in December. Over 150 people participated, both in person and virtually. It was a very interesting event. Beth and Kevin organized, together with the company of biologists, a workshop at Buxted in the UK around effectively communicating bioimage analysis. And this is the list of participants, and I've highlighted the people who are part of our working group. And so you can see that quite a few of our group were there, but also you can see that Katarina was also there, because part of the QCDM working group. She's chairing the QCDM working group. So very good representation from BINA. Out of that workshop, a paper came up that was recently accepted in the Journal of Cell Science. So hopefully we'll have a link to share soon. In Minneapolis, in April, there was the Association for Biomolecular Research Facility. They have their annual meeting, and this was in Minneapolis. And we had a presence there. First, I was part of the fourth light microscopy research group, where we were asking the question, how variable is the analysis of the same data performed by different researchers? And that should come out in a paper quite soon. There was a session by Beth's group on high content imaging. Another session on implementing AI into core facilities, and John, who now is part of the working group, about challenges around bringing AI methods into core facilities. And the three talks were all people from our group. This was also an excellent chance to bridge the efforts between the two working groups. In particular, for example, the light microscopy research group within BRF is now, collaborating with our group, and John also joined us as a result of that bridging. We also had the Pair Up workshop. This excellent initiative. I put a QR there on a recent paper that describes the Pair Up initiative more broadly. And so we had an image informatics focused workshop too, and a number of our members were presenting: David Joy, Simon, Beth, and Kevin. Actually, someone from Simon's group. I apologize. I highlighted Simon because he was part of our group, but the person presenting was Federico Gasparoli. And so we had 37 participants, which is also a great turnout. Our group was also involved in either a scientific or organization committee for two of the upcoming events. One is the I2K in Milan, and the other is the virtual I2K, which is happening just after. I encourage you to check out both of the links. There's still time to register workshops if you're interested in presenting at the virtual I2K. This is another interesting development. The NIH sent out a request for information around software best practices October last year. And our work group submitted a response essentially saying, well, they were asking, what do you think about our guidelines, ours and NIH? And so we responded that it's all very good to have fair software, but you need to fund it to have production quality software. So pay for documentation, usability, incentivizing longevity and fairness of software, and supporting research software engineers. Now, I'm sure a lot of them went in, but the NIH definitely listened, because afterwards there were two calls that were very much related to what we were talking about in the response. So building sustainable software tools for open science, and NIH Research Software Engineer Award, which was great. I mean, it's really great to see that, you know, things can change. So I also wanted to point out that I was linked to this BINA Congress. There is a special issue on frontiers in bioinformatics that is coming out. Everyone is invited to participate. Where this is a list of topics that we are going to be covering, and the first call for participation is going to be in the week of the 7th of October. We're having a deadline for January 30th for just the summary, which were previously the abstracts, and the manuscript submission deadline for April 30th, 2025. So myself and Kevin are the coordinators, and then topic editors, Beth, David, Paul, and John, and Carolyn Miller, all part of our working group. I also wanted to point out a couple of resources that came up in the last year. Everyone is familiar with Pete's introduction to bioimage analysis, but it was also translated in Spanish. And thank you for Mariana Denise, who I understand is in the audience. So thank you very much. And Bioimaging Guide was also translated in Spanish and also Portuguese. So thanks, Mariana Denise again, and thank you, Maria Cruz, for the Portuguese version. Another useful resource is the Halfway to I2K YouTube channel. This was a year ago what would be similar to what the virtual I2K is this year. It's essentially a meeting that happens in the year in between two successive I2K meetings. And it was focused on workshops, and there were 31 video tutorials as part of that. And they're all recorded and available on the YouTube channel. So I highly encourage you to have a look at that. Finally, I wanted to indicate that there's going to be a meeting in November, Northeast Bioimaging, which is happening in November 14th and 15th. And also the hackathon. You already know about the hackathon that Virginia is organizing in Zurich. Yeah, I think I was just about on time, so there's happy to take any questions or.
Applause from the room if that didn't pick up on it. Thank you, Thomas. I think we have time for one question for Thomas as a virtual presenter before we get started with the rest of our presentations. If there's any questions on the working group? Curtis has a question.
Hi, thanks. Great talk. I was wondering about Pete's guide versus the bioimagingguide.org. You said they both got translated recently. I'm a little confused because Pete's on the paper that goes with the .org site, but then he wrote this other thing first. So can you briefly clarify the relationship, or what's the thinking for the progression of these tutorials? If you know.
Yeah, no, I'd love to answer the question. Unfortunately, I don't have a lot more information on kind of the thinking behind the two guides and how they fit together and the relationship with the paper. I think there's a lot of effort in the community to try and bring these resources out. And I guess as part of that is that oftentimes there is a bit of overlap in what is being proposed, but. For this particular instance, I don't have a better answer.
No worries. I can talk to Pete and Beth about it. Thank you.
And Mariana, who is also here, who did the translations of both. So I know, at least from my knowledge, I know one is on QPath and one is on the Bioimaging Guide. So it was those two Jupyter books, I believe.
Sounds good.
And yeah, they can all provide more information. Thank you so much, Thomas. With that, I'll let Beth come up and we'll transition to our speakers. Thank you, Thomas. If you want to stop sharing, I'll start sharing everything on our end. Yes, flash talks first. Oh, did you want to? So our first flash talk is going to be, so these are poster flash talks highlighting some awesome posters that are going to be presented tomorrow afternoon. So we're going to have just some very quick one-slide flash talks from some of the poster presenters to give you a little sneak peek of all the great stuff that's going to happen. Poster presenters, if you want to sort of make your way down this way. But first off, we have high throughput machine learning segmentation with Napari Dev for the generalizable quantification of neurodevelopment from Tim Monco, University of Minnesota.
All right. Thank you. Hello, everyone. Thank you sincerely.
Sorry.
We got to wait for the audio?
Yeah, we're gonna
Okay.
switch for the Zoom audio. Thanks all. One moment. Oh, I'm not sharing. I'm not sharing my screen. Thank you. That was my bad. All right. Sorry, virtual audience. Let's share the screen. Oh, sorry.
Ready? All right. Thank you.
Yeah.
So hi, everyone. Thank you sincerely for the opportunity to share my work with you. I'm thrilled to be here and meet this community for the first time. As I started my postdoc, I shifted from ImageJ to Napari due to its modern sensibilities and the great tools available in Python. But I quickly realized that Napari's plugins often feel disconnected and lack the ability to batch process images. So to this end, I developed the Napari NDev or NeuralDev plugin to serve generalizable image analysis goals. And the widgets can be combined to form a start to finish pipeline. The plugin is designed to accept diverse n-dimensional image formats and preserve necessary metadata. Napari NDev has been used by myself and collaborators to utilize the macro recorder-like Napari assistant for arbitrary and modifiable batch processing, or two, to quickly generate annotations to train machine learning classifiers and predict on batches of images. And finally, three, to measure labels and summarize results based on extractable metadata to have user-friendly data outputs. Overall, Napari NDev is designed to support both novices and experienced analysts because the processing pipeline can be easily designed, modified, and shared. I offer this plugin in the hopes of further development, collaboration, and feedback, so please come chat with me. Thank you very much.
All right, next we have micronuclei detection with vision transformers from Yifan Ren from the Morgridge Institute right across the street, and Vida Agrawala from here at Wisconsin-Madison.
Hi everyone, I'm Yifan and this is Vidi. We are glad to share our research on micronuclei detection with vision transformers. Labeling out the micronuclei manually has always been a difficult task for most of biologists. Thus, we wish to build a machine learning method that can learn and capture the micronuclei phenotypes with prior human knowledge. We built our method upon a pre-trained and self-supervised machine learning method called BINA, which is developed by Meta AI. We have achieved 80% of precision and 85% of recall, which beats the state-of-the-art methods such as CellPulse and MNFinder in micronuclei segmentation tasks only. And also, we hope to push our research one step forward to handle more and different experimental settings. And our post number is 14. We look forward to seeing you there and discuss more. Thank you.
All right, next up, machine learning assisted min-infrared spectrochemical fibrillar collagen imaging in clinical tissues from Wihan Adi, also from University of Wisconsin.
Hi everyone. Yeah, so my poster is about collagen imaging. So collagen is an important biomarker, especially in various cancer. Different cancer stages have different collagen fiber morphology or other properties. And this is the research here is usually traditionally done using second harmonic generation. And using second harmonic, people have shown that there are differences, but the molecular reason behind the differences are still unknown. So here we are utilizing a relatively novel imaging technique called mid-infrared spectral imaging. which contain biochemical information. So and it's a multidimensional data set. So we combine both modalities with help of machine learning, and then with the longer term goal of trying to understand different collagen at the various cancer stages, what are the difference in the biochemical signal. But initially, we are using it for first to segment collagen from the rest of the tissue. Come chat to me. My poster number is poster 7. Thank you.
And last but certainly not least, a postdoctoral training program in bioimage analysis from Suganya Sivagurunthan at the Broad Institute.
Hi, everyone. I'm a part of this amazing postdoctoral training program, which was launched in 2019 by Ann and Beth, with the intention of training my biologists with microscopic experience on bioimage analysis. As a part of this program, we don't just get trained on computational skills or bioimage analysis. We also get trained on time management, project management, and also run a lot of collaboratory projects and deliver outreach activities. The diverse ranking importance given by all of us, the participants, on the different aspects of the program indicates how clearly all the aspects of the program influences our career development. It is not just who are being benefited by the program, but we also develop a lot of materials on bioimaging and image analysis, which are good resources for the bioimaging community. The success of the program is also well reflected by the positions taken up by the former participants of this program. And I'm happy to chat about all the fun that I have in this program later during the poster session. And please come and visit me at the poster 17. Thank you, everyone. And thanks, Beth, for making a part of this program.
And reminder, the poster session is tomorrow. Those of you who are here in person, we are delighted to have you, and we'll be looking forward to showing you some of the cool stuff there. I'd like to next invite up Adán Guerrero from the Universidad Nacional Autónoma de Mexico, who is going to talk to us about extending resolution in space and time with computational nanoscopy. And he is also leading the image informatics working group's sister working group within Latin America bioimaging. So thank you so much for coming, Adán.
Hello, it's a pleasure of being here in BINA. So this time, I'm pleased to present science. Other times I have presented about more societal initiatives, but now let's go to science. Okay, so in 2022, we developed the mean-shift superresolution approach, which is a computational approach that extends resolution in any technology of fluorescence microscopy we have tested. That can be the regular epifluorescence to include to other super resolution technologies. So this is a representation of how MESR works. So it is based on the use of the mean shift approach. That is a Mexican hat. What you can see there is a second derivative operator. And when you process a distribution, no matter the shape of the distribution, you get this inverse distribution that have a minimal of information. So in this approach, we take the negative of the mean shift or the image that is resulting, crop the zeros, and then multiply the resultant distribution by the original to recover the intensity. As you can see, the narrow of the distribution, the distribution become narrower. So after that, so I'm just putting this distribution in here. So all of these steps are called measure of order 0. And then after, what we can do is take the difference between the original distribution and the one of measure of order 0. And what you can get here is a difference of Gaussians, which is another Mexican hat or a second derivative operator. So what you can do then is make the difference of this distribution. Then you correct the inversion and multiply this distribution by the previous one, the previous measure. As you will imagine here, there are zeros, so the tails are cropped. So what happened is that the distribution becomes narrower, and then we do the intensity weight to recover the intensity, and so on and so forth. So something that we are doing is that we can repeat this different orders of measure to refine even more and more. So at a cost that you are losing the tails of the information, okay? So in other words, we are computing second derivative operators, and we are doing an expansion on second derivatives. And every term of expansion is an order of measure. Okay, so if we look at the same process in the two-dimensional, as you can see, we are creating like a step donut, a digital step donut with a minimal of information. And after some an algebraic transformation, we go and sharing the information. And this is very interesting because if we model the having two fluorophores and we approach the fluorophores by the n times their standard deviation, here you can see the Rayleigh limit, the Sparrow limit, which is where there is no minimal of the information, and the measure limit. So meaning that we have in some ways find a way to increase resolution in any image that is created with a fluorescence microscopy technology. So all of that is published, and I will not have time to explain all of it. But something that I want to say is that this is not artificial intelligence. So meaning that we know every step that is going on, and then we cannot speak about hallucinations, but perhaps some artifact like removing information of the tails. Okay, so if you want to know more about how this is working, so there is a talk that I gave in the BINA scientific speaker series, and then I explain a bit more about the deepness of this. So you can take it, check it in YouTube. Okay, so during this process, initially we thought that we invented an algorithm, but we end up inventing a mathematical principle. And this mathematical principle has wider implications because you can adapt it to different technologies. So we got funding from CCI to not only to create the technology, but to put the technology in the hands of the people. So that's why we organize schools in different parts of the world. We organize visiting scholar programs and also a Latin American super resolution meeting. So I will speak something about that later on, but so we also got funding from CCI to create the Napari SuperRes, in which we put measure and other computational super resolution methods together for people to being able to compare what is the best for them. So in one of the schools that we perform in Argentina, so we teach the people to use the Napari SuperRes plugin, and we made a challenge between the methods. So every participant were part of the challenge. The idea was not to demonstrate what is the best method, but to demonstrate what is better in each specific situation. And so we are preparing that work. So later on, we organized, as you can see, it's very close by. We organized the Latin American bioimaging meeting, which was bonded to the super resolution meeting. And in this case, the idea was to spread the knowledge on super resolution microscopy in Latin America. And we got only in the super res meeting 120 participants. 60 of them were presenting a talk, a poster. 50 of them got a fellowship that we provide thanks to CCI. And an amazing thing is that half of the presenters were using this technology in their science. And this is transformative in Latin America because there is not plenty access to high-end instruments. So, but now people can really boost their science through super resolution microscopy. So here I'm presenting something that is going on in our lab. We are extending the resolution in image-based flow cytometry. And as you can see, we can go to high content super resolution imaging because we can extend resolution in a single image. And so we are studying some physiological aspects in the sperm flagellum. As you can see, if you put progesterone, for example, which is an agonist that transforms the cell, the flagellum becomes narrowed. Now we know that that is a process that is driven by actin, and that stops motility. But when you go to high something, you have high problems. And so some of those is what were like, so in this technology, the focus is fixed. You cannot move it a lot, and cells move. So what happens is that you have cells in focus and cell out of focus. Then we use machine learning. In this case, we use the ResNet 50 network to classify and to identify cells that were in focus. Remember, we are interested on the flagellum, and the outcome was very good. But then after we were interested on looking at the flagellum, so we used the UNet to identify, to segmentate the head and the flagellum. And the outcome was not the perfect one. It was a dice of 0.1 0.81 on average. But remember, we are interested on doing super resolution on the sperm flagellum and on those cells. So now we can collect thousands of images and do super resolution. But the next challenge is computational load. So you need plenty computational processing. So, and we went to use other Mexican hats that you can use in the Fourier domain and perform faster computational. So we compare the MESA approach with other second spatial derivative operators, and the results in resolution were the same. But the nice thing is that the times were collapsed, and the things is that it depends in the data set. Always depends on the data set. So what we are doing is doing benchmarking on fair data. So this is about data reuse, and this is very important. And we choose the database of the Japanese guys. Is to create this idea of communication with everyone. So this is the last topic, and we will take like 1 minute. So another application is that you can extend resolution in a space, but what if we extend resolution in time? So that is a fluorescent lifetime imaging microscopy, and what you can do is you can extend resolution with measure, use it as a mask, put it on top of the film image, and then you get but a spatial refinement of the lifetime measurement without altering the lifetime measurement. But what if we go and mess without the lifetime measurement? So we went to the frequency domain lifetime and modeled the entire system and measure it. So that here we have two fluorophores separated in the below the diffraction limit. That have different lifetime, and what you can see here in the bottom is the phasor plot, and that is something very interesting, because instead of having a single lifetime, diffraction could create a blurring on time. So that happened with both lifetimes, but something interesting comes when you have two fluorophores in the below the diffraction boundary. You get a different lifetime, which is not expected. And this is very, very interesting. So what I can tell you is that Messer can correct some of those blurring on time. So here is more or less. It's not fully complete, but we are going on that way. And we already have some experimental demonstration of that. So with this, I would love to finish. Thank you. Merci. Gracias.
Thank you, Don. That was off topic. Okay, just kidding. Thank you, Don. I think we have time for maybe a question or two. Oh, but we'll also have a panel discussion afterward. All right.
Thank you for your nice talk. I am Nicolas van der Voort from SVI Huygens. Can you comment on how conceptually your method compares to SMLM? Yes.
So this is not single molecule localization microscopy. The mean shift theory was developed to find maximum of density. Instead of doing an iterative process to find the dot, so we got the magnitude of the vector. And what happened is that you are in a pixel, and there you reduce the uncertainty, but you don't go to single molecule localization microscopy. But if you are in the region of single particle, single molecule, what happens is that every distribution gets narrowed and the background becomes close to zero. So it increases the precision, the recall, and all of these measurements to do single molecule localization microscopy.
Thank you so much. All right, our last talk in this session before we then proceed to a panel discussion is from Juan Caicedo, formerly of the Broad Institute, but now from right here at University of Wisconsin. And he's going to tell us about towards foundation models of cellular morphology. Thanks, Juan.
Thank you so much. Hi, everybody. My name is Juan Caicedo, and I'm going to talk about foundation models for cellular morphology. And I have to say that in our group, we are also very skeptical of this, working at large scale. And that's one of the reasons why we want to investigate what are the limitations of this type of approach for image analysis. So cell morphology, as it was described before earlier today, refers to making measurements of the cell, of the structure of the cell as we observed it in the images. And basically, for a long time, for more than a couple decades, many of the measurements that we took from images were based on manually engineered features, which means that we have to decide the algorithm and the procedure to process the signal, get some information back, and then use it to correlate that with biological findings. Many of those features require also manual parameterization, and many of those involve areas, shapes, color distributions, and so on. So it also is kind of a involved type of processing workflow. What we ultimately want to do with those measurements is to create perturbation maps that we can observe to study biological variation. So for instance, in this specific map, what we have is one point representing one specific condition, treatment, or perturbation. And we want to know what are the relationships between those, if there is any difference that we can detect with the images. And that's where cellular morphology plays a key role. And there are many ways in which we can create this map by measuring different things on the cells. So for instance, one first thing that we can do is use a cell profiler, or in general, any type of biological tool to measure cellular features. And with that, we can get a certain type of performance. In this specific plot, we see a couple of metrics of how well the perturbations cluster each other in this map. And you can get this type of this type of performance when you use classical features. On the other hand, more recently, we have seen people using more and more deep learning and neural networks. So one way in which you can create this map as well is by reusing pre-trained networks that have been trained with images of cats and dogs and natural images in general. And if you do that for this specific benchmark, we see that the performance drops with respect to classical features. So just reusing neural networks that have been trained out of domain may not necessarily give you the best biological connections in your data. Another thing that you can do is to train your own neural network with your own data. In other words, personalize the model for the type of images that you have captured. And in that case, you may get better performance sometimes, but we have also observed that it's not necessarily the best always. And in this case, according to one metric, which is in the horizontal axis, we get better performance, but not according to the other, with respect to cell profiler features or classical measurements. So what we have observed, however, is that when you train those models with larger data sets, then you may be able to get better performance. And in this case, the blue point is indicating one experiment in which we pulled data from different experiments in order to break certain overfitting limitations specific to batch effects and so on. And in that case, you can actually recover better biological signal in order to create these maps of biological variation. So one of the things that it's more powerful these days is training without labels, and I think we particularly like that type of approach because it gives us the opportunity to do unbiased analysis, just image-based, and just what you capture with the images. And for that, we know that there are several algorithms. So for instance, in this case, this is similar to contrastive learning, in which a couple neural networks look at the same image with different types of views, and they have to solve a problem in which the features match against each other. And notice that in this case, we're not using any biological prior or any labels or manually collected information. It's just image-based, and by solving this type of problem, which is similar to reconstruction and many other type of tasks that we can use to train neural networks, the representation sort of reveals the biological variation that we collect in the images. This particular approach works better with vision transformers, which is a different type of neural network. It's different to convolutional networks, which is one of the most powerful as well for computer vision. And one of the reasons is it has better scalability properties. So when you have tons of data, vision transformers may get better performance as well. And one of the tricks of vision transformers is that instead of scanning the image with a convolutional filter, what we're doing is tokenizing the image or transforming the image into a sequence of patches. And then internally, the transformer network creates or computes the so-called attention across all of the patches in the sequence in order to reveal which dependencies are in the entire image in parallel. So that also expands the range of analysis that we can do in a single image as well. And we have tested those ideas of using self-supervision with vision transformers in a few benchmarks. In this case specifically, we are looking at the LINX dataset, which is a cell painting dataset where the original authors compared cell painting against transcriptional profiling for the task of mechanism of action prediction. And they found that cell painting with cell profiler features, which is in green, it's very competitive with respect to transcriptional profiling. So that was an exciting finding. And what we are showing here is that with deep learning, we can also recover even more signal, especially with self-supervision, which is in blue. And that allows us, again, to create maps or find relationships between biological samples, which is what we ultimately want for different types of biological studies. We have run this strategy as well in different data sets, including the Human Protein Atlas, which has 4 different channels. And using only the images and the self-supervised method, we are able to recover biological variation as well across multiple samples. If we run this again in a different data set, and this one is the Allen Cell Explorer dataset, which has 3 channels, we also are able to recover the cell compartments without any supervision as well. So one last thing that we are investigating is that we know that now we can scale neural networks with vision transformers, self-supervision, but there is still one limitation in order to make them truly reusable across multiple experimental studies. And the problem of making them adaptive to different channel configurations. So in different studies, such as the 3 datasets that we mentioned before, the Allen Institute dataset has 3 channels, the Human Protein Atlas has 4, cell painting has 5 channels. So if we follow the traditional wisdom of computer vision, most of those models are hard-coded to deal with one specific set of channels. In the case of natural images, it's RGB. And the models that we train in one of these datasets cannot be reused in other type of studies. So what we're studying is potential variations of models that can adaptively take any number of channels so we can measure morphological variation in different types of images. For this, we put together a dataset that has samples of these those 3 datasets that I mentioned before with different tasks, and it considers out-of-distribution tasks as well to actually measure how much benefit we get when we train with different experimental data. And there is a model that is kind of promising in the literature, which looks at vision transformer models with channel variability as well. And I refer you to these couple papers that are very recent and are also investigating the same problem. Ultimately, what we want is to scale this, to make it useful across multiple studies. So at the Morgridge Institute and at the University of Wisconsin, we are collecting a lot of data coming from different sources that we are standardizing as well in order to train, hopefully, a model that we can reuse across multiple studies. I want to take a second here to advertise one postdoc position that we have open with my collaborator, Liz Heines. And this is specifically to study 3D imaging, which is a different dimension in microscopy images, and we're going to be working on this soon. So if you know of somebody, please contact us, and we will be happy to chat. So with that, thank you so much for your attention, and thank you to the funding sources and to all the members of my lab who are putting all their best effort to make this happen. Thank you so much.
We probably have time for one quick question for Juan as we're getting set up for our next activity. Thank you so much, Juan. That was delightful.
Hi, Juan. That was a really great talk. I really enjoyed it. I was wondering if you could talk a little bit more about how you foresee building out models that would be more broadly applicable to data sets just in general. Because like right now we have things like CellPose or StarDist that work really well for like general nuclear segmentation. But when you are getting into like having to build out more specific models for like niche data sets, you know, that might not be as broadly applicable or reusable for people who might want to take advantage of those things. So just kind of wondering where you see the future of that going and how that might be built out.
Yes, thank you for the question. I think that's a great question. We think about foundation models that, as models that can be broadly applicable and and useful for different types of tasks. That's one of the reasons why we are collecting, data sets at large scale. I did not present any of the statistics of the, efforts of collecting data, but they come from publicly available sources, such as the Image Data Repository, the Bioimage Archive, and the BBBC or data sets. So when we look at all of them together, we see a wide range of, imaging panels, diversity of cell lines, perturbations, microscopy settings, and even microscopes. So with that, our goal is to have representation of as many microscopy settings as possible to train a model that can accurately measure cellular morphology, being robust to batch effects and so on. So we really think that in order to make it useful, it has to go to a scale where it represents most of the microscopy settings. I don't think, however, that we're going to be able to make it with the current data that is available. So it's going to be kind of a long-term effort to try to represent as much variation as possible. And if your images are not represented here, we are also welcoming, donations of data sets that, you would like to incorporate in this. So hopefully you are going to benefit as well one day from those models that can understand your type of images. I also want to clarify that this is an effort for 2D image analysis. So any other dimensions are not considered at the moment. And I think there is going to be a lot of, interest as well for other type of imaging. Yeah. I hope that we can, have a model that we can use regardless of the image modality. And the reason is there is a lot of, things in common as well. We're talking about microscopy, and there are many principles shared across, all of those. This reminds me a study in natural language processing in which they were looking at 100 languages together, in a single study. And there are surprising, matches, even when the alphabet is different, even when the grammar and the semantics are different. And I think I expect the same, in a data-driven way in microscopy. We're talking about cells most of the time, and many of the patterns, even if they are captured with different fluorescent labels, may have certain relationships that we can exploit and make, more accurate measurements about. Thank you so much.
All right, so we're going to try a little experimental format here. Those of you in the room and those of you online, we would love you to join this Slido. And if you guys want to sit, then we're going to be here for 45 minutes. Or David, you can, but yeah. We're going to be doing some questions about people's in the community's experience with various aspects of deep learning, your hopes, your dreams, the things that are crashing your hopes and dreams. And then we're going to intersperse that with some, based on the responses that you guys give, talking to the experts about what are their advice about maybe doing some things differently or better, or what they see as hope for the future. That all sound okay?
So real quick, my name is David Joy. I'm a scientist at Denali Therapeutics, and I've been doing machine learning for about 10 years. And we're hoping to get some feedback on the different kinds of problems with machine learning, like Beth said. So the first thing is sort of just wide field, like if everything worked perfectly, if all the magical AI comes to pass, what actual problems would we like to solve? Because I think what we've seen is very restricted problems based on things coming out of the computer vision world. But I know as bioimaging scientists, we have a lot of our own special problems that maybe are more specific to us, maybe are things that people in CS have already figured out. We could just steal their stuff, which would make my life easier. So just kind of for a second, think about like, what are your hopes and dreams for the kinds of problems you would like to have a system that could solve? Blue sky. All right, here we go. Cool. All right. So this is kind of the thing that I expected. Oh yeah, it's gonna be hard for you guys to see. So everyone has trouble segmenting their images. That's like the #1 thing that I spend all my time doing. And so it's not super surprising that we see that up there. But there's lots of other really interesting ones up here. Cancer, I like that. That's that we may have to work hard on. But yeah, tracking is a big problem that I feel like is a little bit underexplored in the machine learning spaces. Spaghetti. We've got some noodle themes here. So. But yeah, I feel like as a community, we've kind of gotten maybe a little bit stuck on segmentation as the use case for these things, but there really are, depending on your problem space, a lot of different things that we could potentially use machine learning to automate, to semi-automate, to actually get working a little better. Tell me what can be improved at acquisition time. That would be, what if we had like a pop-up for our users that could do that? That'd be really cool. All right, so I don't know if these are working. Technical difficulties. We haven't tried this. Cool. So yeah, so we've got some basic things that I was trying to think of in terms of like how would we like to use deep learning? Like if someone gave you like a tool to do deep learning, if someone told you like, oh, here's $1 million, go do deep learning, whatever that means, how would you actually like to use it to help your work, to help your trainees, to help your core function better? All right, cool. I'll pop up the answers. Oh yeah. So everyone would like to just have it like work. push button, receive machine learning. But yeah, no. So I mean, I think this has been a huge problem in the field so far, is that we have model builders who are really, really good at using PyTorch or TensorFlow or whatever, but they don't talk to the imaging tools at all. And then we have people who use Fiji who can't take advantage of all these tools. So I think definitely there's a huge opportunity there to bridge these things. But I think a lot of the issues, especially with commercial tools that I've used, is that they won't let you actually play with the model yourself. I don't know if anyone else has run into this issue. So I think also thinking about ways of integrating training loops into our tools so that we can take our data set and not somebody else's data set or ImageNet or whatever, and actually apply it to our real data, which has its own real quirks that we know about and maybe can take care of. I'm actually impressed by how many people want to write their own code, though. That's great. I fully support that. it's tough, but it can be learned. Yeah. And it seems like I was actually expecting a little bit more of I don't need to use it. I feel like if I asked this survey 5 years ago, everyone would be in that last bucket. So this is actually exciting to see that there's some enthusiasm here. Do I want to restart? Sorry, guys. One more time, I promise. Okay, so of all the tools out there, which ones have you actually used or given to a student to use or like helped a student use or have running in your core somehow? So we saw segmentation was the biggest problem. So there's lots of segmentation models out there. I gave like a couple, but anything segmentation flavored. Do people use ChatGPT? I know everyone has been telling me to use ChatGPT, but I don't know if I have actually like used it to do anything useful. I mean, there's lots of other things. People were saying tracking. There's tracking models out there. Are we actually using them? Are we using these classifiers or feature extractor kind of things? Or something else? Is there anything weird that people have used? Yeah, so I mean, like large language models, people love that as an interface, especially I feel like people who are less computationally savvy, people who aren't even used to using ImageJ will still go onto ChatGPT and be like, how do I use ImageJ? And ChatGPT will say something. Hopefully right, but sometimes. But yeah, segmentation. Image classifiers. I'm actually, and we'll open up to discussion in a second. But that's an interesting one that I was expecting to be a little lower down. So, that'll be cool to see. And then people are trying it for super resolution. That's bold. Okay, cool. So that was like, what did we try to do? But like, if somebody gave you like $1 million grant to do AI, or I don't know, $10 million or whatever, which ones would you like to do? Like, what do you wish you could actually implement in your course, in your lab? Yeah, cool. So now I feel like ChatGPT is a little bit further down, which maybe that is lower barrier to entry, right? So like, I think a lot of the problem here is that the things that we would like to do feel hard to implement. And then something like ChatGPT, well, it's there for better or worse. So, I mean, I guess I'd like to have our panelists up and maybe we can talk a little bit about sort of challenges getting some of these things to work.
Yeah. So Juan and Adon, did anything surprise you here in terms of what are people doing? What are people not doing? If you want to come up to the chairs.
Yeah, I would like to start with the main topic, which is segmentation. So we most of the time want to solve this bottleneck that we have in image processing, but at the end we in some way realized that, and we saw some examples today, that the experts have to be there to train the models. So I wanted to open the discussion to see how much extent the expert will bias machine learning models for segmentation. So we have seen that experts not often very agree with their segmentation. So how much of that we are putting into the segmentation process that everyone's want to have?
I think it's great to see, interest in the segmentation problem specifically. I guess it comes more from a need, right? Like segmentation is usually the first step towards processing your images, regardless of what type of structure you're interested in. So I guess it is not surprising that, we need better segmentation tools, and that, includes any type of analysis. Cell segmentation is probably one of the most studied problems, but there are many other structures that we want to segment, right? Like including the specific compartments or structures or, you know, the filaments and stuff like that, which becomes increasingly harder and harder as you aim for a specific structure. So I find that particularly interesting. It's also one of the most long, you know, the longer effort in segmentation, the image analysis problem that has had attention for the longest time is segmentation itself. I think it has more than 50 years of research, and we are still, you know, in the deep learning revolution, and it's not solved. So probably one of the main limitations for why it doesn't really, you know, happen yet, it's because it relies on supervision. Segmentation is one of those problems that requires a ton of manually annotated data, and it's hard to collect the images in the first place, and then someone annotated them. So it seems like it's going to be a problem that we have for a little while.
Hi, Chloe from University of Ottawa. I have more a comment from what we heard a little bit this earlier from Virginie. And I was surprised that there was actually no mention of it in the questions. So you talk about the segmentation and so on, but very often you see that the segmentation is cannot be done properly because of the image quality. And I would actually love to have AI, deep learning, being able to actually improve the post-acquisition processing in order to optimize the image, to improve the image, so after it can be segmented correctly. How many times we tried softwares and it doesn't work, so after trying on the ImageNet once, and the person will say, well, you image actually with maybe a 10x, you need a 20x with a higher resolution. Here you didn't, we cannot segment these cells or this tissue because suddenly you have a drop of the fluorescence and we cannot make, you know, a continuous line, stuff like this. So actually, before the segmentation, I would love to have something that can tell me what kind of tools I need to use. Do I need to do a Gaussian blur? Do I need to denoise? Do I need to do this before going to the segmentation? I don't know if someone thought about this before.
Yeah, working in a facility where you are, people is like you, we are always in the frontier of, what if you go and collect the experiment again? Is that possible or not? So in that sense, so as an image analyst, sometimes I see these kind of problems that the quality of the image is not good enough. It has compression, it has very little signal. And as experimentalist, I always ask, can you repeat it? Yeah, sometimes it's possible. Sometimes we spend much more time analyzing data that is challenging than remaking the experiment. So, and there are plenty examples of that. In the cryo microscopy is pretty clear that you need this boundary, and sometimes it's better to collect again the data. So I pretty agree with you that if we go and perform collaboration with companies, with developers that recommend the experimentalist to get the better parameters for the sample, that might improve the pipeline before image analysis. And I will change the terms that I will recommend to change the term there instead of going and say, how many of you have used super resolution? How many of you are interested in doing restoration? I think that is much more important. But the problem of restoration is that you end up dealing with hallucinations. So, yeah.
Yeah, I wanted to, the magic denoise button is definitely something. And again, there's a lot of people working on denoising networks and things like that, and they work, but designing ones that will
only give you real denoising and real magic denoising and not hallucinate things is just an incredibly big challenge. And how much do you want to sort of base your results on something that an AI cleaned up for you? I mean, people are making them, they exist, but it's an open question. How much are they safe to use? And it's going to probably be extremely problem to problem dependent.
Yeah.
Yeah.
Yeah. So yeah, so there actually are image deep learning and non-deep learning algorithms that are specifically designed to detect noise level, magnification, find out of focus things. They work sometimes. But yeah, so there are definitely tools that will sort of like quality check your images, but of course they're all based on assumptions too about what are minimum blur levels, what is the minimum magnification that will be somewhat problem dependent. And similarly, there are actually tools. There's a really cool tool called Napari Omega, where you type into Napari like a text box, say, I want to analyze, like unblur my image for me, and it will propose a set of discrete image processing steps that will like write out basically like a simple like image processing pipeline for you to filter whatever the AI thinks to filter your image, which whether or not that's correct, that's an interesting thing, but that plugin does exist. So people are working on that. But yeah, that's a great pipeline builders as like LLMs is an interesting and like hot topic in research right now.
All right. We have some more questions. So these are great discussions, but yeah, just want to make sure we get through getting more information from you folks.
Do you want to do one?
So yes, how much do you agree with this statement? There are conceptual barriers that keep me from using deep learning. So things that you could use deep learning more or better if you understood the theoretical concepts.
Yeah, that seems about right. I think anyone who's claiming that they understand deep learning or even machine learning perfectly is probably, I mean, maybe there's a couple of them at Google, and Meta, but beyond that, I think we're all still kind of struggling to wrap our brains around these programs and how they work and how we can best take advantage of them. So yeah, I think that's having community resources and education out there. And also like thinking about like the format of the education that we want to provide to people is a huge issue in the field right now. So instead of conceptual barriers, now it's like practical barriers. Like I don't have enough computers, or I have too many computers, or I don't know how to use all these computers, or why is my data not on the computer I need it to be on, which is my problem all the time.
Or installation sucks.
Or yeah, how do I get this software to work? Somebody please get this software to work. Yeah, no, so this is the other current struggle I think in the field is basically like there's a lot of plumbing involved in getting these things working even slightly, and getting them working in production is kind of a nightmare. So, I mean, there's definitely platforms online, like we saw some of them this morning, that people are trying to use, but it's not always perfect. All right, one more. If you haven't started, or if you started and then stopped, what do you think was the biggest barrier?
And maybe while folks are typing those, Juan or Adon, do you want to mention something that you've either had a difficulty with yourself or noticed your collaborators having any difficulty with getting started doing deep learning?
So can you repeat the question, please?
What are difficulties that either you personally have had or people you work with have had getting started with deep learning?
Yeah, well, it's all related to the ecosystem where you are. I mean, the biggest scientific discoveries are related to people, but indeed there is an emergent phenomenon of the community. And that is something that we can see in our communities that sometimes, for example, in Latin America, the community is not ready to really assimilate the content that is out there. And the biggest need that I foreseen is education, but the education has to be tailored to the communities, to being able to the education to penetrate. So one of the experiences that we have had is that people came to help us, to ask for help to solve the problems, but they are very, very far away to learn how to solve them in the time that they need. So I think that the better way to approach this problem is to create and to push much more energy in education to spread the knowledge in the community, and that knowledge will create this momentum that will help us to solve most of these problems.
I think, well, with respect to the two previous questions, and like how, how much we need to understand the deep learning methods or the machine learning algorithms in order to use them in practice. I think that more important than the specifics of the algorithm is the methodology of how you apply those. I believe that, not even the creators of those algorithms sometimes truly understand how, this is working. And it's definitely a non-deterministic type of system in which you're driven by probabilities and statistics and so on. So it's also data dependent. But regardless of how, because it's becoming more and more complex. So regardless of how exactly it works, I think the important part is more like how we use them. And I don't think of those, methods or models as tools yet that we can, just apply in any conditions. We really need to be careful about, how you apply them and how you validate them. I think that's the most important step in the entire process. How you convince yourself using the scientific method that what you're obtaining out of those models is consistent with reality, is consistent with what you would expect. So I think that's where I feel like there is a little bit more disconnection when we work with collaborators we want to apply deep learning. They are very enthusiastic and they see a lot of promise because they have tried everything, like in the micronuclei segmentation problem, for instance. I remember our collaborators tried every possible tool out there and manually configuring parameters. And there was always a trade-off of we're going to get more or less depending on how you do that. And when they want to use deep learning, the problem is that it's not necessarily a tool that you can just tweak. It's really a methodology in which you have to think about, what data you leave out for validation, what data you annotate, what data is used for training, and so on. So I think that's the major barrier, like learning how to test and how to validate those methods is what we really need to, probably discuss a little bit more as well.
All right, maybe we can throw to the audience for a question.
This is Xiang from University of Utah. So my statement a little bit off the AI topic, but it's related to what Chloe mentioned. So like last semester, I was being assigned to teach a 1-hour class for the graduate student. So the topic is like how to take a good image. Okay, so I kind of like scratch my head. It's hard to start it, right? So, but anyway, so the first words I talk to my student is the magic happens before you bring the sample in, right? If you have bad sample, basically it's hard to get good image. So, so you don't think the magic will happen if we just, have all the good tools or AI tools without good sample. So that's the thing you just keep in mind. Yes, science is hard.
I would love to comment, to follow your comment. So we have been giving a lecture of image analysis for up to 10 years in Mexico. And the first 2 months of the course is about microscopy, about data collection. And then after we go into the fundamentals of image analysis. And one of the slides that I always put in my lecture is that before doing image analysis, try to understand the fundamentals of the physics, of the maths, and of the biology that represent the data that you want to analyze. Because that is a very big decision that you have to take. So at the end, people is looking for a scientific question, and image analysis is not magic. It's just another tool that is used to reach that goal. So I think that is something that has to be embedded in image analysis course in everyone, which is data collection and data management and so on and so forth.
Hi, Ronnie Kurmaker from Harvard Med. My question is slightly off from the topic we're discussing right now, but more on the development side of deep learning models. Now, in the LLM world now, there is a discussion about whether we create one massive model that is general and enough that it can answer any question. And on the other side, the other thing is more of a mixture of agents where you have tiny, small models. Considering the vast diversity in biological images, where do you see us going, and what kind of model do you think would work the best? Kind of one model to rule them all, or more on the small models that come together?
Yeah, I think, well, I think this is a great question, and my guess, because I don't think I have the answer really, but my guess is that for certain type of microscopy studies, we are probably better off with a single model that we can share in order to, get the most out of the images. And I think that is probably what's driving, forward computer vision in general, because they have a very standardized type of image type, and they collect more and more data on that specific niche, if you want. One of the issues or challenges with microscopy is that, microscopists and biologists are very creative, so they keep inventing more and more image types, and that's going to be an issue for, this model that rules everything. I think we definitely will need also models that are specialized for new types of images, multiplexed images, time-lapse, 3D. It's a really, diverse and unique universe that we study. And I don't think we have data sets that cover all of that space or that spectrum. So my guess is that this is going to be more of a combination of both, like there is very recurrent type of imaging techniques that we use in the lab. And for those, we probably will have a model that can help us in basic tasks like segmentation and so on. But every time we, push the boundaries with new studies, new type of images, we're going to need to personalize, customize, and specialize certain models for extracting useful information from those.
I would love to comment. So I think that we scientists have to look back into the story of science. So if we look to these biggest equations that describe nature, let's say that we are speaking about the Navier-Stokes equation. If you try to use that equation, you cannot learn much about the specific system. You have to define boundaries. And that is something that I foresee in these technologies, that a general model will help us to give a general picture of how the things are working out, but at the end we will have to define boundaries to make this technology to really take over and to give us a meaning of how nature works.
So, oh, sorry, just, oh, God, you go ahead. Okay, just to speak briefly about the LLM space, because that's also kind of an interesting development. There's been a huge push towards generalist models in LLMs, but what's fascinating about them is that when people test them on actually focused data sets, they're not very good. And so there's actually been a huge amount of literature in the last few years about taking these generalist models and creating essentially like small models like barnacles that stick onto the sides of them that modify them for particular data sets. So there was a really beautiful paper that just came out last year of taking essentially ChatGPT, the model that underlies it, and training it on a diverse set of medical data sets. So there was one for like clinical radiology, there was one for like certain kinds of oncology. And basically each of those little mini models was much, much better at answering the questions in that data set when it was latched onto the sort of main generalist model. And so I think there's a potential to like, even if we do end up with some sort of like general purpose, all singing, all dancing image model, there's still going to be need for specializations for, again, the vast, vast creativity of microscopists.
Roland Nitschke from Freiburg. So it's just comments. I think we should avoid using AI as a tool which makes out of crappy things good stuff. Yeah, and that is currently the danger. Also, when we look at the development of instruments, there is more or less all new instruments come out with somewhere mentioning we are using AI. So in the end, actually, what counts for microscopy is samples, quality of the instrument, and quality of the image. And then you should use AI and not the other way around, produce something with AI, with maybe a crappy instrument, and then, yeah, come to some result. I think that would lead us in the wrong direction.
Garbage in, garbage out is certainly a principle in computer science as it is everywhere, but there's definitely also room for thinking about how we rescue precious samples that would otherwise be totally lost. Are there cases where having an imperfect recreation of something is better than not having it at all? And so I think there's a lot of interesting ethical questions around how you do that and then how you disclose it. The thing I always tell people in image analysis tutorials is nothing is actually really wrong if you write down that you did it.
So from chat, Kara Winter was asking, what's the best tool to use use deep learning for tracking nuclei in like 3D over time, like for light sheet images? And do you have any suggestions there? And also maybe how do you find the right deep learning tools for your for your biological questions? How do you know what's out there? And
that last one is really hard. There's basically a new one once a week. So if you're making new deep learning tools and they are open source license, we would love to have them on forum.image.sc. So at least there's one place where everyone can find them. I've heard good things about ULTRAC and about BTRAC in terms of specific recommendations. I don't know if you guys have other specific recommendations, but yeah, the state of the art doesn't last very long, but the cell tracking challenge is also a good place if you want to look at performance of tracking algorithms specifically. There's actually benchmark data sets for things.
Yeah, that's basically my recommendation for models in general is don't find the hot new paper that just dropped on arXiv or bioRxiv. Find the review with like a big challenge that tests like 10 models head to head. And then whatever one they say is the best, try that one. And then if that one doesn't work, try the second best one, and so on until you find one that actually works for your data.
Especially if they aren't introducing their own new model in the review.
Yeah, and I think that this highlights the importance of benchmarks. I don't have a recommendation for cell tracking specifically, but I think the idea of having benchmarks that you can use to compare and so on, it's important. And if there is a benchmark for your task, maybe you can, design one test for your own problem. As far as I understand, in cell tracking, segmentation is a core issue, and the state of the art is, based on accurate segmentation. So it kind of comes back to, having good segmentation algorithms and so on. But how do we know which one is the best? And I think that benchmarks is the answer.
Yeah. Donate your data to us so we can make benchmarks, please.
Just to come back to the discoverability point, there's bioimage.io, the bioimage model zoo, which is like, I think a good starting point for discovery. Nobody said it, so I just thought I needed to say it because it's great to talk about how to tell which ones are the best, but how to know they exist. Okay.
Yeah, that is another great place. Florian, you will murder me for not having said it first, so thank you, Curtis.
Hi, Eric Waite from Alphys again. I have more of an anecdote here. I was in a registration challenge back at NIST a long time ago, and there were 3 winners. One was a very, very complex algorithm, used optical flow, and it took about a day and a half to run this registration challenge. And it was able to get precision down to sub-pixel accuracy. I was second runner-up on something a little bit less sophisticated, but just did some convolutions and was able to get pretty darn close. And then the winner was, the trick question from NIST itself was, you didn't actually have to change anything because they made sure that their stages were very well calibrated. So if you just placed all the pixels down where the stage said it was, you were done. So I guess that's just a tale of caution of sometimes you don't have to jump for the most complex, the most sophisticated deep learning things. There are many other classical or handmade features that you could try first. And there's plenty of forums to help you on some of those. And then when you need that precision, then you start going up. But don't start for something that's so complex, so precise right out of the gate.
Yeah. I love that anecdote because my favorite thing to tell students is if your images are good enough at the beginning, you can just threshold them. Like that'll work as long as you take a really, really good image.
Everyone make sure their metadata is accessible.
Yes. All right. Unfortunately, we're at 1 minute remaining, so we may have to put other questions to the speakers at lunch, but thank you to all of the speakers so far this morning, Virginie before the break, Juan Adan, all of our poster presenters, Thomas remotely, and everybody else. It's been a wonderful session. Hope folks enjoy the next Tech Bite, and then we'll head to lunch.